my pleasure today to introduce today's speaker, Dr. Jim Murray from the Department of Biological Sciences at Cal State University East Bay. And I see Friday Harbor Labs. Jim, I pulled a little bit of biogeographical data off your website. So Jim got his bachelor's at Cornell in 1988 and a PhD at University of Washington in 1994, and I guess set some roots at Friday Harbor at the time. Postdoced at UC San Diego, was a professor at the University of Central Arkansas, starting in 1998, and then moved to East Bay. I don't know if it was that before or after the name change.
Before. Or no, after.
After. So
after the name
formerly
change.
Hayward State, and then later Cal State East Bay.
Right.
So I've known Jim since 2008 when he came to East Bay, and Jim's been a friend of Moss Landing Marine Labs since then, bringing classes down here, and a very avid participant on many of the cruises that we used to do, the class cruises on the Point Sur and deep sea exploration. Those days behind us for now, but maybe coming back if the new regional class vessel gets funded and we get to be one of the operators. So I was gonna say that, you know, it's such an unfair thing that the speaker has to watch everybody eating, and the speaker's like too kind of wound up to enjoy the food, but here he's chowing down here. So very relaxed speaker. So Jim's discipline or area of research is in neuroethology, so the study of behavior from a neurological and physiological perspective. And an invertebrate zoologist, the sea slug is the organism he uses. You may or may not know that sea slugs are an important biomedical model in neurology for the reasons that I'm sure Jim is going to tell us. I wanted to tell Jim one thing, though, about Friday Harbor Labs, which is it's an amazing place. I love to go there. I was there as a student, and I taught there as an instructor. Jim teaches the neuroethology class there. But what he should know is that Moss Landing Marine Labs has plans to develop a academic village with housing for visiting faculty and research space, and we're hoping to recreate some of the spirit that you find in summers at Friday Harbor Labs locally, a place where CSU faculty and scientists from around the country can come and spend their summers kind of in a similar way. I know it'll be hard to pry you away from Friday Harbor Labs, but getting it out there. So without anything further, let me invite Jim to talk about the sea slug Tritonia tetraquetra, neuroethology, sensory function, and chemical ecology.
Excuse me. Okay, yeah. So yeah, that was great. Who's responsible for that? Oh wow, that was super good. So yeah, I study this sea slug with the unpronounceable name. It used to be known as Tritonia diamedia, but you know, when people go through old collections, they find things and then they figure out that something has precedent. So that's its new name, which I actually like to say, Tritonia tetraquetra. And so yeah, primarily a neuroethologist, and but I've had sort of the freedom to pursue other kinds of pursuits, so into chemical ecology a little bit and a couple other things too. So tell you a little bit about what I'm doing up there at the Cal State campus. And you heard that little song that I played there. That was a song that was composed by some University of Washington students, obviously sea slug brain people. So this is my slug. It is shown slightly larger than life size, but life size adults are 2 handfuls. I've seen a 3 handful slug, so they're really big as far as animals, as far as nudibranchs go. And so you can see their cute little faces here. And oops, there's the laser. So they've got this structure called the oral veil. So the mouth's underneath there. The oral veil is chemosensory touch, chemosensation, and touch. They've got these olfactory structures, the rhinophores up here, which are involved with smelling prey odor, prey and mates. And then they got these nice little brachial gill tufts. So I've been studying a lot about their sensory mechanisms, how they determine where they want to go, and kind of the motor mechanisms of how they actually do it. And so the main reason we study these animals is because of this brain. So this brain is about 6 millimeters wide, about 7,000 brain cells in it, and people have counted. The larger cells here you can see are quite large. Some of the larger cells are up to 1 millimeter large, and they are 80–90% nucleus. So they're polyploid, like 2 to the 12th or something, like 5,000 copies of their genome. Not sure why, presumably to generate enough mRNA to generate enough protein to maintain an axon that can be 10 microns wide or maybe 20 microns wide, and then go out 20 centimeters out to the body. But it makes it relatively easy to get electrodes into these individual brain cells. And then the other thing about their brain cells is they're relatively stereotyped in their position, such that these cells here that you can sort of see outlined in orange are so-called metacerebral giant neurons. They are found apparently in every species of gastropod, right? So what's that, 100,000, I don't know, something like that species. They always are serotonergic. They always modulate feeding. And then there are other cells here that are found that are re-identifiable across individuals of the same species, sometimes across genera, sometimes in the entire nudibranch group. Also, you can see some white cells here that are peptidergic. Some other cells here have unique neuropeptides in them, and people have been doing, colleagues have been doing complete transcriptomes of every cell in the nervous system of various nudibranchs. So we're making some progress there. You can see the nerves radiating out here, and each of the nerves has been sort of mapped out to various parts of the body. So the initiator of this research was Dennis Willows. He was my PhD advisor, and so he had an animal in the lab that was kind of a pet. He was actually assigned to work on something else, but when this pet died, he cut it open, saw that picture of the giant brain there, and noticed that he could make out some of the brain cells with the naked eye. So presumably very large, and no one at the time had recorded from inside a brain cell in a brain that was still attached to an animal, and of an animal that was still moving. And so what he's doing there is doing that right now. Intracellular electrodes in the brain, the animal sort of hanging hanging by its head, and then can engage in natural behaviors as long as it doesn't involve moving its head. And so he was the first person to do intracellular recording from a moving animal. So he developed this little technique here where you sort of open up the head and you immobilize the head, you put a platform underneath the brain, and that helps stabilize the brain in order to keep the intracellular electrodes in. And this is what that technique looks like. You can see that guy, and you notice the posture there is pretty good. They don't like it when you cut into them, but maybe 15 minutes later, maybe half an hour later, they look pretty normal. They seem to be okay. And so here a little closer up, you can see the lobes of the brain there with the intracellular electrode going in. And here you can see some of these cells are whitish, peptidergic cells, and these pins used to sort of immobilize the brain. We use tungsten because the animal apparently has a magnetic sense. So we try to use non-magnetic materials so as not to distort the Earth's magnetic field. Oops, sorry, went back one, go back one. Okay, so like I said, some of these cells are re-identifiable across individuals. And so you see little numbers there. So this cell here, pedal cell 5, has been shown to respond to magnetic field rotations. And so their consistent properties allow us to continue experiments across individuals and still be looking at the same, essentially, genetically determined neuron. Okay. We've probably identified a couple dozen of the cells as individuals and probably a couple hundred as functional groups. But that still leaves several hundred or thousand that we haven't quite figured out. The smaller cells that are in between all these spaces here are full of smaller cells that we haven't identified uniquely. But again, the cells are large enough that under microscopy you can identify which cells are expressing which peptides or which neurotransmitters. So I've been doing a little bit of that neurotransmitter kind of mapping to look at functional connections there. One of the first behaviors that Dennis Willows was interested in studying was this escape behavior, which he had observed. Pycnopodia here, the sunflower star attacking the sea slug, and it goes into this sort of random undulations, dorsal flexion followed by ventral flexion. And one of the questions that was fundamental back in the 60s was, how does the nervous system prevent incompatible behaviors from being activated at the same time, right? And so dorsal contraction and ventral contraction are basically incompatible behaviors. So he was interested in how do they do that. And so after hiring a dozen different people and a couple decades go by, they came up with this, okay? And so it shows these two neurons, the dorsal swim interneuron and the ventral swim interneuron, mutually inhibiting one another. So that's what prevents them from activating dorsal and ventral at the same time. And so that was the first time we understood how the brain or how nervous systems organize themselves to prevent incompatible behaviors. And we've since found this reciprocal inhibition is illustrated in our breathing control and our walking. Almost all rhythmic behaviors have some aspect of this mutual inhibition in it. And then they've also worked out the entire neural circuitry for that escape swim. They've modeled it, and so they're pretty sure they know the vast majority of the neurons involved with that behavior. But what I got really interested in is their orientation to water flow. It's one of the most obvious characteristics that they like to orient to water flow. You put them in a sea table, they end up hanging around the water flow intake. And so you can sort of see the crud in the water heading that way, and you see him crawling up that way. His oral veil, that's a pretty typical posture, sniffing up here. And then the oral veil, you'll notice that there are some larger papillae here on the edge that are sort of brushing the ground. Later on, I'm going to show you some results from that, but we think those might be another olfactory organ specialized for sniffing the mud. And so I got interested in this orientation to water flow behavior as one of their principal orientation mechanisms. And so I took them in the lab to sort of characterize that behavior, time-lapse them because it's boring watching them in real time. And so I start them at 4 different orientations with those yellow T's there. And so I start them at these 4 different orientations, and they always end up crawling upstream. So it's a very strong reflex for them to crawl upstream, and presumably because upstream is where you get all your smells from. Okay, and so over the years we've learned that they have this strong attraction to water flow. They're also attracted to the odors of food. They eat these sea pen corals. They are also seemingly finding mates by odor. There's also evidence that they can smell predators, and they've developed the ability to orient relative to the Earth's magnetic field. And we're working on mechanistically how they do that. Where is the sensor? How is it connected to the nervous system? So what I'm really interested in ultimately is that question of integration, right? When an animal has incompatible or information, do you compromise? Does winner take all? And with a brain that has only 7,000 neurons in it, we think we can get to the question mechanistically of how does a brain make a decision, right? And trying to figure that out in vertebrates is much more challenging. So a little outline of the brain here showing kind of the functional organization of the nerves. So we know what nerves are going to those rhinophore structures. We know what are going to the oral veil and to the mouth. A little optic nerve that goes to an eye that is so regressed underneath the body wall that it probably can't form images anymore. But we know it's physiologically functional. And then we also know where these various nerves from the pedal ganglia are going to sort of control posture and crawling. So it's a nice system to work in with lots of background information. So it also makes it a little more, makes you reluctant to jump species for fear of what you won't know in the new system. Okay, and so I focus right now on this pedal ganglion, which I said is innervating the foot, mediating crawling. And so this neuron, my PhD advisor, Dennis Willows, had done that in semi-intact experiment. He stuck an electrode in pedal 3, stimulated it, the animal bent like that. Stimulated the other side, the animal bent like that, and said, aha, it's involved with turning. And I was like, that is a facile conclusion, and I don't believe it for a minute. You've got an animal that's, tied up and hung there, and you didn't actually do it while they were crawling, so I don't, that's too simplistic. It's probably untrue. So I took that as my thesis project. And so over the years, we've done various experiments. This is what it looks like when you stimulate that cell in an animal that's not hanging by its head. And so this is work by an undergraduate that I worked with. And then it's in time lapse again, sort of sped up there. But it shows you they lift that part of the foot, right? And the idea is that if they lift that part of the foot can't be involved with propulsion. Therefore, the other side of the foot will propel them towards the flexed side. So that was kind of our working hypothesis. And so this shows recording from that neuron, inject electrical current into that neuron to cause an elevated firing rate, which persists for a little bit after the stimulation. The black bar is the duration of the stimulation. And then down here, showing that same 10-second stimulation, and then the red here plotting the spike rate going up and down, and then the blue showing the motion of the foot and the fact that the foot lifts and stays there for a good long time. Takes a minute, well, not even more than a minute, to eventually fully relax back there. So they're slow animals. Takes a long time to do these experiments. But this shows you kind of the time course of that response. And the time course of that response was measured with this little fiber optic device here that measures light reflection without touching the animal. Okay, so showing that neuron does something that kind of looks like turning is one thing, but obviously one would want to record from the neuron while the animal is turning. And so that involves doing something not intracellular electrodes. They have a tendency to break off if you don't stabilize them. So what we did is adapted a technique that we learned about from another scientist. This is basically just a little piece of wire. It's 25 micron thick wire. So it's thinner than human hair. And basically, you take off the insulation and then basically turn it into a little brand, essentially, glue it, place it onto the ganglion. This ganglion has been sort of pulled out of the water, right? So you notice the animal's brain's so dorsal, you can just pull it out of the water, blow it off with some air, put the little brand on there, and then glue it in place with super glue. And believe it or not, that works. And so this is work from my former grad student, Roger Redondo. Okay, there's a little glue patch there. And so that insulates it so you can put the animal back in the water. Okay, and so this is what it looks like doing that kind of recording. In this case, so each spike here is a spike from that one neuron, and then these tiny spikes in the background are adjacent neurons that are electrically further away. But we can tell that this one is the largest spike, and we did simultaneous intracellular and extracellular to prove that that was the same neuron. Okay, and so what you're showing here is an animal crawling around in a tank, showing its spike rate going up, its spike rate going down, and so the cell seems to burst on and off, and you can see the little spikes there. What Roger set up now is a 3-camera system. So you've got a camera on the left, a camera on the right, camera, I think in this case, below the tank, videoing through the plastic. And then you run water flow, so they'll have a tendency to crawl upstream while you have a fine wire glued onto the right pedal 3 neuron and the left pedal 3 neuron, and record that all on one system. And so that's what you're going to see here is a view from the right, review from the left, and then a view from below. And in this case, the water flow is coming from above. So let's see what that looks like. And okay. Next. Oops. I think I got to click on that. Oh, shoot. Oh, that was my favorite movie, too. Yeah. Sorry, I'll have to dig that up later. But basically what you're going to see, what you would have seen here, whoops, what you would have seen here is this animal turning up in this direction and the wire sort of trailing off the animal. So this graph is showing that firing rate of that neuron, okay? So left pedal 3 neuron, and when the animal is engaged in a left turn. So right, this is a sea slug pointing down, not a fish pointing up. Sea slug, and then the white arrow is where the water flow is coming from. So we basically put them in the tank, forced them to be sideways to the water, and then recorded their neural activity. And so spike rate going up and down, up and down, up and down. Fastest it got to is like 2 spikes per second, but that's fast for slug. And then when it got oriented to the water flow, we then grabbed it and twisted it again to make it turn the other direction. So now we've got them this way, water flow coming from the right, spontaneously it'll turn to the right and eventually ends up pointed that way. So the dark blue line says you were 90 degrees with flow on your left, and then you oriented to flow, and then you had 90 degrees flow on your right, and you oriented to flow. So when turning left, lots of activity in the left turning neuron. Activity on the right, or turn to the right, less activity in the right turning neuron. So I have to admit that Dennis Willows was correct with his simple experiment, and we didn't have to do this big complex thing. But showing that there's a direct correlation between natural behavior is a stronger bit of evidence. Still, one would like to know, is that the only neuron involved with this? Maybe this neuron's just correlated with the turning behavior. And so, oh, sorry, this showed the right neuron should do the same thing, right? So the right neuron should have elevated activity during right turns and less activity during left turns. So shows that that's symmetrical like that. We don't really know what the significance of the bursting is, but obviously that elevated activity is not constant. It sort of comes in these little bursts. Okay, and so our kind of model for how this behavior, this turning behavior is doing is we've got these turning neurons right and left. We know that they burst kind of simultaneously, okay? But we also know that if you stimulate one and look at the other one, it doesn't respond. If you stimulate the other one, look at the other one, it doesn't respond. So they're not connected to each other. But they are synchronous in their bursting pattern. So because of that synchrony in their bursting pattern, we think that there's a separate neural element sending a signal to it. And then that signal is a patterned output of some sort. So a central pattern generator that elevates the activity in both neurons simultaneously. But then we have to ask the question, why, when water flow is coming from one way, does it elevate the activity? And so The idea is that flow modulates the synaptic strength of each of these differently, right? So if you have left to right flow, it enhances this synapse. If right to left flow, it enhances that synapse. So we're still trying to work out all that neural circuitry, but we have identified the nerves that carry that sensory information. Okay, and so that's carried through these nerves called the lateral cerebral nerve 2 branches. So just doing nerve cutting experiments to prove that that sensory information is coming from there. Okay, but like I said before, showing that the activity of those cells is correlated with behavior doesn't prove that it's the only cause of that, or even any cause of the behavior. It doesn't prove causation. So to test causation, what we did is injected an anti-sodium channel chemical. And so that lidocaine derivative here is a water-soluble lidocaine derivative. And then it'll spread throughout the entire cell here, mixed in with a green dye, so we can see the extent of the injection, and essentially turn off just those 2 neurons out of the 7,000, and then basically glue the animal back up and throw them in the tank. Okay, and so pre-surgery shows you that same kind of turning from sideways towards water flow, from sideways towards water flow, from those 4, 45-degree directions, showing that they're mostly going upstream, statistically clustered. And then post-surgery, the animals are going more or less straight. Okay, so you start in this direction, you keep going that direction. You start in that direction, you keep going, basically straight lines, or kind of a lazy turn in the wrong direction. So we took this as good evidence that those neurons are necessary for turning behavior. And in fact, they may very well be sufficient for turning behavior. The other thing that we tried to do is shock the electrodes when we had those glued in place. Shocking the electrodes also forces the animal to turn. The only problem with interpretation, though, is as you're shocking that, you're probably activating that one neuron. It's hard to be sure that you're not activating other neurons. Okay, and so this was that sensory nerve cutting experiment I was telling you about. The cerebral nerve 2, when you cut that nerve here in its entirety, they're totally disoriented. So then the next thing I did is went to the next branch and said, well, I wonder if the information's coming in through the lateral branch or through the medial branch. And so when you cut the lateral branch, when you cut the medial branch here, cut the medial branch, the animals are oriented very well. Okay, and so that basically is average direction for 4 different slugs. When you cut the lateral branch, very disoriented. One animal was statistically significant to the 45 degrees off thing. So maybe there are some variations in where that sensory information comes in, but primarily coming in through the lateral branch of cerebral nerve 2. So we're still pursuing that and trying to figure out what the nature of the receptors are and exactly how those synapses get modulated. Okay, so like I said, we're interested in water flow reception. We're also following up on questions of magnetic orientation and on odor. And so I want to show you some of our odor experiments as well. So like we said, Tritonia preys on these sea pens, and so sea pens, Penatulacean coral, and it pretty much exclusively preys on them, at least in its adult life. There are some examples where there's a habitat where they might have 2 species of Penatulacean there, but again, it's still another Penatulacean. And then these corals are thought to be toxic to most animals, right? And so the nudibranchs specializing in preying on toxic prey. So this is kind of a long video. I don't know if I'll watch the whole thing, but basically you can see it getting close to it with its rhinophores out, knowing that it can smell something. Once it gets close enough, it starts rearing up and lifts its foot up like that and starts positioning itself. The reason it has to be very careful is that entire sea pen, and you can see one in the background there, that entire sea pen can withdraw into the sand. and it can do it in like 20 seconds, which is faster than a Tritonia can really move very fast. So what the Tritonia is trying to do is orient its oral veil and orient its jaws so it can take a bite, but it knows it's only going to really get one bite, and then the animal will probably withdraw. And so it wants to touch it very gently. And then I'll call your attention to these tentacles right here, which are larger than the rest. You see they're a little meatier. Those are the ones that I said are sniffing at the ground. They tend to drag them on the ground. But right now they're sort of positioning, touch, touch, touch. They're blind, right? So they're trying to like figure out where I'm going to bite this thing, and I don't want to startle it too much. Okay? And then I guess just I'll ask Jonathan, put Jonathan on the spot, if he can remind me what the little black guys are in the background. Yeah, I always think I should ask someone, then I forget. Taneids. Okay. But yeah, numerous. Okay. And so use olfaction to get in the neighborhood. Use touch to position themselves, and then use it's crazy. And then use taste to know that it's the right species. Right. Yeah, they got to be really careful, right? You know, you don't want to spend an hour sneaking up on a sea pen and then just miss it, right? So they're slow animals. And then the video is only 2 minutes long because I run out of air and have to leave. But it makes 2 attempted bites. It misses both times, right? And then I ran out of air. But did you see one when I was looking? I think it's coming up on an attempted bite. There it is. Yeah, missed. Clean miss. But at least it didn't withdraw, right? And but they have these left-right jaws. They'll grab a piece, they'll tear it off, and swallow it. Eyes could be useful. And like we said, their eyes are there. Their eyes are functional. We've recorded from their optic nerves nice action potentials, but they are so buried underneath the tissue they certainly can't form a clear image, and they don't have any obvious visual behavior. Yeah, he's just going to miss again. And there we go. And I say he, right, but I mean he, she, it, right? They're simultaneous hermaphrodites. So showing a couple other shots there. Again, that extra large oral veil tip there, which has a special sniffing organ in it. Yeah, you can see it right there and right there. And then this one actually got a bite there. So they're eating this animal even though almost nothing else will, right? There's like a couple sea stars and a couple slugs that'll eat this thing. No fishes, no crabs. Okay, and so that looks what their rhinophore looks like. Some SEM there, and so in addition to having kind of diffuse sensory cilia on their surfaces, in here, in kind of deeper into the folds here, there are fields of motor cilia that end up actively pumping water through the clavus of the rhinophore. Okay. And so here's a live Tritonia, and you see the little beads getting pulled in there. So it's a pretty substantial suction, and so really should increase the effectiveness of sniffing. And you can see it gets entrained from quite a distance away here, right? With the viscosity of the water, it's sort of sucking in from millimeters away. Okay, and this shows you some green dye getting sucked in and then blown through the folds of the clavus there. So, and also you notice here, the posture they keep these things out, right? They're sort of stretching, right? Which makes me think they are attempting to get out of the boundary layer and maybe get more rapidly changing olfactory information. And then at the same time, you've got this structure down here, and you can see the little indentation there. That's the groove that has the cilia in it. This grooved organ here is dragging along the bottom, which makes me think it's doing the exact opposite, getting chemical information that's not changing rapidly out of the boundary layer. And so, you know, very different kinds of strategies for sensory organs. another undergraduate collaborator. Abigail has been working up on this work now, and she's really interested in kind of diversity, and so looking at not only this rhinophore, but the rhinophore of related nudibranchs, which all have various weird rhinophore structures, and you always must ask yourself, why do all these rhinophore structures differ? All they are is surface area. Why should it matter? And apparently they're not just surface area. They are pulling water in different directions. These are Dorid rhinophores, and they're bilaterally symmetrical. And what I've noticed is that the Dorids will actually rotate their rhinophores to point in different directions. And since they are actively pumping from the anterior to the posterior, they can actually point at where they want to sniff, right? And so we're sort of looking into the functional significance of that. We're also really interested in why does Tritonia pull water from high? Why do these animals pull water from the side? Does it have an ecological correlate with the types of animals that they're hunting? So we're still just getting started with that. Okay, and then another interesting question is how does that sensory information coming in from these olfactory nerves, the cerebral nerve 1 is known to be the olfactory nerve, how does that kind of sniffing smell get to turning neurons to affect turning? And so that's what my grad student Samantha has been working on, and we've been doing some confocal microscopy to look at overlap between the sensory pathways and the motor neuron. And so she hasn't finished that work yet. Okay. Another interesting question is how does all this information get integrated together? Orienting to water flow and to odors and to magnetic fields. How could they possibly be related to each other? So what you're seeing here is a time-lapse video of an animal, and then this little orange hockey puck here is a neodymium magnet. These are those super strong magnets that really distort the magnetic field, probably in this whole area here. And then you're noticing water flow is basically going left to right here, and we're showing water flow with the fluorescein. And then what we did is we laced the water flow with C-pen odor. So crawling upstream, I like C-pen. And then what we did is we turned off the water flow. And you saw I threw a little box around it to sort of immediately stop the water flow. And so it was crawling upstream happily, smelling the C-pen, and then the water flow turns off. So they still have olfactory cue there, but the olfactory cue is kind of everywhere at this point. But without the water flow, they start searching randomly. Okay? And so what we're really interested in is if you deny them water flow information, would they then rely upon magnetic guidance? So we were kind of hoping that when we stopped the water flow, the animal would crawl exactly straight, despite the fact there was no water flow cue anymore. But it didn't turn out that way. And so what we're really interested in is what they use this magnetic sense for. We know they orient to the magnetic field, but we don't know ecologically why. Okay? And so these shows the track of all those slugs, okay, crawling from this direction, basically starting from right or left, turning upstream towards the odor that's coming from over here. And then we sometimes have a magnet here. We sometimes have a control puck there. And so we were curious about when the control puck was there, does it cause the behavior to be different than when the control puck's not there? And we really haven't found too much difference there. But what we do know, the obvious behavior you did see, is that you release a slug, it turns towards the water flow, and then as soon as you turn off this is a different experiment, where you turn off the odor and you leave the water flow, they start turning back and forth. Okay, so water flow is an orienting signal by itself, but then when you steal odor from water flow, they give up and they start searching around for where that odor flow came from. Okay, and so this is just quantifying that increase in angular velocity, right? So if before that they're going pretty much straight, and then they increase their angular velocity right here at 0, where they start turning right and left. this is kind of average data. So some of them are turning a lot, right? So that individual data shows a huge turn, and then the average shows elevation that's significantly higher than this. Okay. So my colleague here, Russell Wyeth, showed that in nature, the animals will orient to water flow, and especially so right before a feeding or a mating encounter. So basically, he set up videos and watched them all day, and then retrospectively said, oh, that one was crawling that direction, and then 5 minutes later it mated. Oh, it must have been following an odor plume. These animals here, basically measuring what direction they head off in after the encounter, apparently not water flow guided. So apparently it's an interaction of water flow and odor that motivates them to crawl upstream. And so we call this odor-gated rheotaxis. And like we said, we hope that as animals were crawling upstream, they'd smell this odor. And then if water flow were to change direction before they get there, right? Water flow is pretty turbulent, and these guys are crawling pretty slow. So if water flow changes direction, you could give up and follow another plume, or you could just keep crawling straight. But you know, crawling straight's not easy when you're blind, right? And so Ken Lohman is the one who showed that the animals will orient to magnetic fields in the absence of all other distractions. But he had some speculation as to what that might be good for. But again, we haven't proven anything about what it's good for. But our hypothesis is this so-called orienteering hypothesis that if you had a magnetic sense, you could keep crawling straight and follow that magnetic bearing. So my colleague here, Sean Kane and Andres, have viewed various microscopic tissues. So this is a transmission electron micrograph of a portion of the foot. And you see this chain of electron-dense particles. He wasn't able to determine that chain was magnetic. You can just tell they're electron-dense. But you can see they're kind of made out of a crystalline structure there. They're about 100 nanometers wide, possibly consistent with being a magnetosome, a chain of single-domain magnetite. And then Andres here liquefied the Tritonia, blended them, and then put it in a vial with a magnet in the bottom, and just then took out the fluid, hoped that there was some magnetic material in the bottom, and took a look at it. And so you see things that are similar kind of size. This is like 3 singles there, and this might be a ring of a bunch of them, or a sphere of a bunch of them. You know, around the same size here. So we know those are magnetic, but we don't know where they came from in the body, or maybe they're contamination. But we're continuing to pursue that about how they respond to magnetic fields. So, right, so we're hoping that if they had that magnetic sense, they'd be able to keep on going and make it to their target, even though water flow changed direction. So what we did is we went up to a field site, and oops, oh, sorry. I should have showed this earlier, right? I told you this gray graph before. Some of them had a hockey puck, a real magnet. Some of them had a control puck. And so magnetic in red here, no magnet in blue. And I think you can see there's no real apparent difference. So basically we concluded that you take away odor, they panic and start running around in circles. And so we're trying to figure out, still trying to figure out what that magnetic sense is used for. So here it shows statistically indistinguishable how much they turn with a magnet or without a magnet. So what we did is we set up a field site again, set up cameras, and videoed them basically all day for a couple weeks. And so set it up at this field site near Tofino, British Columbia. And that's what the field site looks like. Lots and lots of sea pens there. High density of slugs as well. You can see why vision's not good in the short distance there. And you see all the sea pens oriented one direction because they're passive flow veins. Okay. And so we grabbed a bunch of slugs, threw them in a pile, and then videotaped them in these with sets of 3 cameras all pointing in the same direction to sort of get redundant information. Okay. And so what we set up are more of those neodymium magnets. Okay, each of these little flags here has one of these really strong, these are almost the size of a golf ball. They're really dangerous. If you can get them near your phone, it could really mess them up. And so then we put this thing, aligned it to the Earth's magnetic field so we know which way was north. And then we set up these little water flow indicators with fluorescein baked into plaster of Paris. And there's the magnet right there. And then have them crawl around, right? So we predict that if an animal were crawling in a straight line and it was crawling towards a sea pen or a mate, some of those animals, just by random chance, will try to do that, and the water flow will change direction. And by random chance, some of them will have water flow change direction and crawl by a magnet. Some of them will have water flow change direction but not crawl by a magnet, and we've got different predictions for those. So we're basically continuing to analyze, I don't know how many thousands of hours of video tracking all these different slugs and all these different magnets. We've got some preliminary data. But yeah, here you can see all 6 cameras on the boat running up with these cables here. Okay, and so what Russell showed is that as the animal approached some of those neodymium magnets, not significant difference in their turning angle, and then suddenly, right here in that range, significant difference in the turning angle, and then back to not different. Whatever, that doesn't seem consistent with the hypothesis. But anyway, the magnetic field extremely distorted there. So we might have some preliminary evidence indicating that they are using magnetic sense in natural habitat. My undergraduate student Celia here is setting up a semi-intact preparation here. Inside this red wires here is a magnetic coil. And so inside that coil, we can change the direction of the Earth's magnetic field. And then these are data from similar experiment by Morgan showing the overall activity here of that, what I told you was an olfactory nerve. And again, we don't know where the magnetic sense is coming from. It could be coming from any number of places. But showing here the spike rate, and then she rotated the field 60 degrees and got an elevated spike rate out of that nerve. So we think that the magnetoreceptors could be in that part of the body, but they might be in every part of the body. There's no reason they can't be everywhere. Tissue is mostly magnetically transparent. And so I wanted to show you a little bit about that experiment, that organ that I was telling you about, this ciliated groove here. And you can see kind of the bumps of cilia in here. And so that's this structure here, pointed downward and sucking water up in this direction. So we're interested in does the rate of transport change when they have different odors in it? And again, maybe, but we haven't seen any really good, strong evidence for that. So we don't know that they're paying attention to this as a sensory organ. It just sort of looks like a sensory organ. And this shows what it looks like when you stick material in it, and you see it being transported out of there. So that's being transported from the tip, basically towards the mouth. So it's a very effective suction device, and it just looks like it's got to be useful. So again, we're trying to figure out what it might be useful for. Okay, and then I said I'd show you a little bit about our ecology, right? And so I've been focusing on the sea slug Tritonia here, but we said that the predator, or potential predator of the sea slug, is this Pycnopodia sunflower star. And oh, incidentally, the reason she's got her fingers there is because she's got an evertable stomach. She can pull right out of her stomach. This is FHL invert ball. But remember, Tritonia has a perfect, I think 100% effective escape response, so it's only a potential predator. I don't think Tritonia has any real predators. And then Tritonia, of course, chasing these sea pens, trying to eat them, and then the sea pens themselves eating plankton. So we got a nice little food chain there for our group costume for invert ball. Okay, so like we said, this Tritonia is eating this slug. Tritonia is eating the sea pen, and it's been published that there's a chemical in it called tylosartanone that has been shown to have an anti-acetylcholine esterase activity. And anticholineesterases are the same target as nerve gases and organophosphate pesticides. They're basically the most effective killing things we have. And so here's our liquid Tritonia. Nathan here has extracted various chemicals from them, and so we're trying to figure out, are these chemicals actually chemically anti-feedant, and then physiologically, what are these chemicals doing? So basically, you can contaminate artificial diet with Tritonia or with Tylosarcus, the sea pen, and you can see in each case, it greatly reduces the amount of feeding done on it. So we haven't done that experiment with just the extracted tylosartanone because it's hard to get more than a few little micrograms. But basically, Tritonia appears to be an anti-feedant, and Tylosarcus, the sea pen, appears to be anti-feedant against crabs. Here's a little hermit crab taking a ride on a Tritonia there. The other thing that we tried to do is we were asking, does the sea pen toxin affect the nervous system directly? And so here we're recording from the nervous system, sort of monitoring overall activity out one of the nerves, and then applying sea pen extract, and it caused elevated activity. So we think the nervous system is sensitive to some chemicals in the sea pen. We don't know if they are less sensitive because they've adapted their proteins, or if ordinarily these chemicals don't even make it to the brain because they're detoxified in the gut. And we're continuing to follow up on that too. The other thing that if they are using, being exposed to an anticholinesterase, we should find out where are they using acetylcholine as a neurotransmitter. This is an acetylcholinergic neuron in the brain, and you can sort of see its three-dimensional structure there. So we know that there's a number of cholinergic neurons in the brain that could be potential targets for this toxin. And what we're doing now is sort of probing for the acetylcholine esterase gene, trying to locate that in the body, and also using an artificial substrate for acetylcholine esterase to see if we can locate that in the ganglia. Oops, what happened there? Oh, that's not good. Anyway, questions while I'm fixing this? Shout them out. Find this. Which side is it on? We did get a prize for that one. We did get a prize for that one. I cannot find whether this screen is on the right or the left of my screen.
Oh, that's coming.
Oh, there it is. Finally found it. Okay. Don't send. There we go. Okay, and get that back. Okay, it's to the left of my screen. Get that back there. Yeah, so we got a prize for that costume that year. Let's see if that's it. Good, that's it. Now it's opening the crashed file too. It should be okay. Yeah, I think so. I think I need the crashed file. Okay, where are we here? Boom, 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 boom. And boom, and boom. Okay, sorry about that. Okay, so ultimately I'm kind of interested in both ecology and physiology of these critters. One thing that's really interesting, the observation from a number of years ago, is the veliger larvae of these animals will not settle on Tylosarcus. Okay, so the adults eat Tylosarcus, but the veliger larvae won't metamorphose on the sea pen, but it will metamorphose on these more gracile penatulaceans that are found deeper. And so, and the other thing that's interesting is in these habitats, I typically find these slugs here spontaneously generating out of the mud at this size, right? And so you look at size class and it's just big, 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 nothing. And so I'm like, where are the small ones coming from? So presumably migration. And since we know that these grassal sea pens tend to live deeper into softer sediment, we're thinking they settle down there, they grow up on these soft, low-flow sediments, and then maybe they migrate towards the shallower water, presumably because with higher water flow, there's more plankton eating and more biomass there of sea pen or something. And so maybe that's what they're using their magnetic compass for. Maybe the adults don't use it at all, right? So they use it as juveniles to get to sea pen beds, and then they're done. But we're still working on kind of ecologically. Obviously, ecological works underwater is difficult and slow. Okay. The other thing that I noticed is that this one site here where I was collecting my sea pen, my slugs for many years, basically I went back to it in 2013, and all the sea pens were gone. And that was like a football field of sea pens. And so you always see Hipposteria around there. They eat sea pens. And I'd seen them come and go, but I'd never seen them eat them all. And so maybe Hipposteria ate them all. I don't know. But it was basically, it was between, I think it was the fall, August and December 2012. They were gone. So it's just 3 months, essentially. So I'm really interested in what causes that. I don't think it's the slugs themselves. Crap. But I went and explored other sea pen sites. Another site lost a very large field of them between 2003 and 2010. I wasn't diving there a lot. I found a YouTube video that showed lots of sea pens here in 2008, and then I went in 2012 and they're gone. So yeah, I'm concerned and kind of curious what's causing that. The other thing that I told you about, the eyes don't seem to be used in vision, but we also are starting to see evidence of circadian rhythms that may be mediated by light. So here you got a little slug who just took a nap there. You saw he was crawling around, and then he sort of tucked in his oral veil, pulled in his rhinophores, and just sitting there for a while. So we think that it might go through sleep, and we know that in vertebrates, right, sleep is involved with memory consolidation, stuff like that. And again, if we can figure out kind of the physiology of sleep in the 7,000 brain cell slug, we might be able to provide some insights for the function of sleep. Okay, there are their little eyes there, little black little spots there, deep, not too far from the brain, right? Basically here-ish and here-ish. And so yeah, I hope you agree with me that Tritonia is the most interesting slug in the world, and they're fascinating. They can do escape response. They have no predators. They're amazing. And so I want to acknowledge all the support of my students and funding agencies, and open it to any questions. Thanks for coming out. Yeah?
What role does the staticist play sort of in?
Yeah, we haven't looked at that too much. So they have motile cilia in their staticis. So the little otoliths are moving, or statists are moving around all the time. And one of the interesting behaviors we notice is the animal will crawl up a wall and then sort of lose its grip and fall, and then lay on its back there. And an animal on its back can and does sometimes right itself in 15 seconds. But other times it'll sit there for hours. and so I don't know if they're paying a lot of attention to those statists. And I wonder if this particular species lives on flat, sandy bottoms. It's just evolved away the righting response. It's like, what do I need it for? I'm never going to fall over. If I do fall over. So I think what's happening when they fall over like that is they're still crawling. They don't notice it. I haven't encountered anything yet, but I'm still crawling around. That's my guess. So I think this species does not pay much attention to its statuses. But obviously other nudibranchs are much more responsive. So we have gotten interested in sort of studying that too. We haven't really made much progress there.
When they swim, they lose contact while they're in puppy.
Yeah.
Land
And
any way, so
yeah,
why
they can
why
right
would they
themselves.
lose
No, that's true.
orientation?
No, that's true. They can right themselves, but you will see them sitting on their back for hours at a time. Yeah, maybe that's it. Maybe it's just a sleep thing. They just happen to fall asleep on a wall and fell over. Yeah, yeah. But I think they have no predators, right? And also people of like you said, they're being used as a neurological muscle. Mostly aplesia, right? Aplesia is used by hundreds of labs worldwide. Tritonia not so much. But aplesia is used as a model for learning and memory. And so naturally, some of the scientists looked for learning and memory in Tritonia and found no evidence, right? And trying to find classical conditioning or associative learning in an animal means you have to get into their head a little bit, know what they think is important, right? To get them to associate two things, you have to kind of know them a little bit. And so maybe we don't know them well enough to know what to associate, or maybe they cannot learn, which is really unusual for any animal to not have any associative learning whatsoever. Paramecium can do associative learning. Classic condition, they only got the one cell. And so I think Tritonia is perfect. It doesn't need to learn because it is perfectly adapted to its unchanging environment, right? It's cold. So yeah, I want to hear the argument against that. But no escape response, no food limitations. I've never seen them like in a place where there's a CPEN every half mile or so. They're always surrounded by food, and they have no predators. So I think they're perfect. They don't need to learn. But yeah, tell me about it sometime. They've evolved perfectly to their environment that doesn't change.
Well, so what does the phylogenetic neighborhood look like? I don't know much about epistemic phylogenetics, but how many species in the genus and where do they live? And
no, that's true. Yeah, there's not too many, but there's a few, and we are making some progress on that with the phylogenetics too. Any other questions? Yeah.
So those children looked oddly familiar to me.
Yeah.
Were they here last year at open house?
Yeah.
So James Murray is on the governing board, and he brought his kids here to open house, and yes, they had an awesome time. They looked so excited when I met them.
Yeah,
I remembered looking at them here. So thank you, James Murray, for.
Yeah, they are basically junior microbiologists. They spent like almost every summer of their lives at a marine station, and so they know a lot about marine biology, and they're obviously really enthusiastic about the invertebrate ball. Right? If you haven't been to the invertebrate ball in Friday Harbor, right? It was founded down here at Hopkins, so it's a real hoot.
your little video clip of the water motion on the rhinophore.
Yeah.
I noticed, it was kind of getting sucked down the middle, but then it was really cooling at the base and, and just kind of sitting there.
Yeah.
And actually some of it was being kind of recycled in.
Yeah.
Was that just in the lab environment? Or?
Yeah, I think that's just in the lab environment because ordinarily, and that's another thing that I didn't show today is, ordinarily that overall bulk flow is going to be pulling things, out of the neighborhood. But so what I'm hypothesizing is that two things is that it increases the volume that it's being, increases the volume that it is able to suck, right? So instead of just sucking what's going by you here, you're sucking something in a larger volume, and you're increasing your temporal resolution, right? Instead of having something stick on your rhinophore for 3 seconds, you're blowing it off in 1 second or something like that. So that's the idea, is that they suck it down, down here. They suck it down here, and then the regular bulk flow will sort of blow it off at that point.
So do you have any kind of broader models for rhinophores? These people may not know these are just a few of the many other, types of rhinophores that you would see.
We found a couple that don't seem to have any active pumping on them. Hermesenda doesn't seem to have any active pumping. We studied a pleurobranched as well, and no active pumping. Yeah, we looked at those 5 species.
The ones that are pumping, why the diversity of morphology? Do you have ideas about that?
Not yet. Not yet. Yeah, that's a really fascinating question. Cool. Thanks for coming. Appreciate it.
That was great.